Good morning. My name is Pinky Devi. I am from 6th semester Botany Department. So the topic that I got is mRNA versus tRNA. So first of all, in this topic, we have to understand first of all what is RNA. So RNA is basically a single-stranded molecule which is present in the majority of the living organisms and viruses. It is made up of nucleotides, which are ribose sugar attached to the nitrogenous bases in a phosphate group, which includes adenine, guanine, uracil, and cytosine. So let us have a complete studied comparison between tRNA and mRNA. So first of all, we will start with mRNA. mRNA is a single-stranded RNA which carries genetic information from DNA to ribosome, which is present in the cytosol, and it is used as a template for synthesis of protein. mRNA is furnished with genetic codes or messages as a sequence of codons, and each codon consists of triplet of bases. There are three main types that is present in RNA that is included in protein synthesis. So the first one is messenger RNA, that is mRNA. The second one is transfer RNA, that is tRNA. The third one is ribosomal RNA, that is rRNA. Now we'll talk about the structure of mRNA molecule. So it is a long polymeric molecule made up of nucleotides. mRNA is a long single-stranded molecule consisting of nucleotides which is attached by the phosphodiester bond. It contains 4 nucleotide bases, that is adenine, guanine, uracil, and cytosine. Mature RNA consists of the following regions. So this is the structure that I have drawn for mRNA. This one is 5' cap. This region is 5' UTR, and this region is coding region. This is 3' UTR, and this region is poly A tail. So now we will discuss all these regions one by one. It is made up of codons, that is the triplet of nucleotides. Each codon codes for specific amino acids, and codons in mRNA get translated by ribosome to amino acids in a polypeptide chain. The coding region begins with the start codon, that is AUG, and ends with any of the stop codons, that is UAG, UAA, or UGA. A portion of the coding sequence may also have a regulatory function. Untranslated regions, that is UTRs, they are present at 5' and 3' regions before and after of the coding region respectively. Untranslated regions are present before the start codon and after the stop codon. They have a role to play in gene expression. They are involved in the stability of the RNA, the efficiency of translation, and mRNA localization. Now is 5' cap. A cap of methyl guanosine triphosphate cap is present at the 5' end, and the last one is poly A tail. Polyadenyl tail is present at the 3' end. So now we'll talk about tRNA. Transfer ribonucleic acid is a type of RNA which helps to decode a mRNA, that is messenger RNA molecule, into a protein. tRNA functions at a specific site of the ribosome during translation, which is a process that synthesizes a protein from an mRNA molecule. Now we'll talk about the structure of the tRNA. The mature tRNA consists of the 3 hairpin loop structure which give it an appearance of the 3 clover leaf structure. The tertiary structure is like the L shape, and the foldings which is present in the tRNA is because of the hydrogen bonds. The folded structure of the tRNA molecule is present because of the hydrogen bonding which is present between the complementary bases. Now we'll talk about the structure of tRNA. The mature structure of the tRNA comprises of 3 hairpin loop structure which give it an appearance of the 3 clover leaf structure. The tertiary structure of tRNA is of L shape. So now we'll start the explanation of tRNA structure with the exception. It is formed by the base pairing of 7 to 9 nucleotides of the 5' terminal to 3' terminal. The 5' terminal has a phosphate group, and the 3' terminal ends with a specific sequence of CCA or CCA tail. Acid attaches itself to the 3' hydroxyl group of the acceptor arm. DHU loop or D-arm. D-arm has a stem of 3 to 4 base pairs and it ends in a loop called D-loop, as it generally contains dihydrouridine, which is a modified nucleotide. Now the anticodon loop, present on the mRNA for the amino acid it carries. These unpaired bases of the anticodon loop pair with the mRNA codon. Each codon is identified by the specific tRNA. Now the T-loop. The T-arm consists of a stem of 4 to 5 base pairs and a loop containing pseudouridine and a modified uridine. This portion of the tRNA is called the variable arm or variable loop. It is present between the T-loop and the anticodon loop. Its size varies from 3 to 21 bases. It helps in the recognition of the tRNA. Let us summarize the topic mRNA versus tRNA by pointing out the main differences which I have explained earlier in the video by pointing out these main differences. So first we will talk about mRNA and side by side we will discuss tRNA as well. So first, mRNA is a specific stranded molecule that carries genetic information from the DNA to the nucleus to ribosome in the cytoplasm for protein synthesis. And in tRNA, tRNA is a clover leaf shaped molecule that transfers amino acid to the ribosome during protein synthesis. Second, it contains codons, which are 3 nucleotide sequences that specify particular amino acid. And for tRNA, it is written, it contains anticodons that complement the codons on mRNA, ensuring the accurate insertion of amino acid into the growing polypeptide chain. For mRNA, the mRNA acts as a template for protein synthesis, where each mRNA molecule codes for a specific protein. And in tRNA, it is written that the tRNA acts as an adapter molecule between mRNA and amino acid, ensuring the correct amino acid is added to the protein chain. So the fourth point is it is synthesized in the nucleus and then transported to the cytoplasm for translation. tRNA is found in the cytoplasm and it plays a crucial role in protein synthesis by bringing the correct amino acid to the ribosome. mRNA undergoes post-transcriptional modifications such as capping, polyadenylation and splicing before being translated into a protein. And here, tRNA molecules are highly stable due to their secondary structures and are reused multiple times during protein synthesis. For mRNA, it is relatively short-lived compared to tRNA and is constantly being synthesized and degraded based on the cell needs. And for tRNA, the amino acid attachment site on tRNA is called the 3' end, where the amino acid binds through an ester bond with the tRNA molecule. For mRNA, mRNA is essential for the process of transcription, where the genetic information ended in the DNA is transcribed into mRNA before translation occurs. So I have drawn the structure of mRNA and the process where DNA develops to mRNA, and the process is transcription, and from mRNA it converts to protein, and the process is known as translation. And here, and in tRNA molecule undergoes specific modifications such as base modification and the addition of the amino acid to ensure their proper functioning in protein synthesis. With this, we have come to an end of this video, and I would like to thank Sir SMS for giving me this opportunity on making a video assignment on the topic mRNA versus tRNA, and I would like to thank my friends and family for gathering information on the topic. Thank you.